You're holding a bone fragment. It is smaller than a penny. It came from the pinky finger of a girl who died roughly 75,000 years ago, inside a limestone cave tucked into the Altai Mountains of Siberia. You have never heard her name because she never had one. Not one that survived. And until 2010, nobody on Earth knew her species existed at all. For roughly 300,000 years, a separate line of humans lived, spread, and eventually vanished across a territory that may have stretched from frozen Siberia to the tropical coastlines of Southeast Asia. They were not Homo sapiens. They were not Neanderthals. They were something else entirely. And the only reason we know about them is because one fragment of a child's finger ended up in the right laboratory at the right moment in the history of genetic science. 1 decade earlier, and the technology to read it did not exist. One careless field assistant, and the bone goes in the discard pile. The margin between discovery and permanent ignorance was that thin. This is the story of the Denisovans, the humans the fossil record almost completely forgot. The ones whose DNA is still running through the bodies of billions of people alive right now. And once you understand how we found them and what they left behind, the story of human evolution that you learned in school starts looking like a rough draft that somebody published way too early. If you're enjoying this so far, hit the like button and subscribe. It genuinely helps. Drop a comment telling me where in the world you're watching from. I actually read them. The cave where everything changed sits about 6 kilometers from a village called Chyorny Anui in southern Siberia. A hermit named Denis reportedly lived there in the 18th century, which is how the cave got its name. Locals had used it for shelter for as long as anyone could remember. Livestock waited out storms inside. Archaeologists had been digging in Denisova cave on and off since the 1970s, pulling out stone tools, animal bones, the occasional Neanderthal tooth. The site was interesting but unremarkable. Just another deep cave in a mountain range full of them. Dozens of papers had been published. Nobody had found anything that rewrote a textbook. In 2008, a Russian archaeological team led by Anatoly Derevianko recovered a tiny bone fragment from a sediment layer dating to between 50,000 and 75,000 years ago. It looked like it came from a young girl, probably between 5 and 7 years old. The fragment was so small and so visually unremarkable that it could have been mistaken for an animal bone and discarded. It very nearly was. But the team bagged it, labeled it Denisova 3, and sent it west to Leipzig, Germany, to a laboratory that had spent 20 years preparing for exactly this kind of moment. At the Max Planck Institute for Evolutionary Anthropology, a Swedish geneticist named Svante Pääbo had devoted the better part of his career to a problem most of his colleagues considered unsolvable. Ancient DNA degrades. With every passing century, it fractures into shorter and shorter fragments, gets swamped by bacterial contamination, and becomes progressively unreadable. Conventional wisdom in the field held that DNA older than a few tens of thousands of years was simply gone. Pääbo disagreed. He spent years developing decontamination protocols, extraction techniques, and sequencing methods designed specifically for ancient, damaged genetic material. By the mid-2000s, his lab had already achieved what most researchers had called flatly impossible. They had sequenced Neanderthal DNA, a full genome, pulled from bones tens of thousands of years old. It was one of the most significant achievements in the history of evolutionary biology. When Pääbo's team ran their analysis on the finger bone from Denisova cave, they expected a routine Neanderthal result. The cave contained Neanderthal artifacts. The region was well-established Neanderthal territory. Everything pointed to a boring confirmation of what they already knew. It was not boring. The mitochondrial DNA extracted from that bone was so different from both modern humans and Neanderthals that it sat on an entirely separate branch of the human family tree. A branch nobody had ever detected before. Whatever this individual was, her lineage had diverged from the common ancestor she shared with us and with Neanderthals roughly 1 million years ago, and then gone its own way for hundreds of thousands of years without leaving a trace anyone had recognized. In March of 2010, Pääbo's team published their findings in the journal Nature. They had identified a previously unknown species of human, not from a skull, not from a skeleton, not from a jawbone with identifiable features, from a speck of bone you could lose between couch cushions. They named the species after the cave, Denisova. Now sit with how deeply strange that is. We had an entirely separate species of human, walking the earth for a span of time that dwarfs recorded civilization. living across what appears to have been an enormous geographic range, interacting with other human species in ways we are still cataloging, and the sum total of physical evidence that they ever existed fit inside a sandwich bag. As of the most recent published count, the confirmed catalog of Denisovan fossils consists of that finger bone, a handful of teeth, a small skull fragment, and one lower jawbone, found in a completely different cave, thousands of kilometers away. You could hold the entire known physical record of the Denisovan species in a single cupped hand. Everything else we know, and we know a startling amount, comes from their genome. Pääbo's team managed to sequence the Denisovan genome with a level of coverage and precision that would have qualified as science fiction 2 decades earlier. From that genetic data, researchers began reconstructing what Denisovans actually looked like. A 2019 study, led by Liran Carmel at the Hebrew University of Jerusalem, used patterns of DNA methylation to predict Denisovan skeletal anatomy. The results suggested individuals with dark skin, dark hair, wide skulls, long dental arches, and protruding jaws. Their teeth were enormous, significantly larger than those of both Neanderthals and modern humans, which implies a jaw built for serious chewing force, and a diet that probably included a lot of hard or tough plant material. When the first Denisovan tooth was found in the cave back in the year 2000, the researcher who examined it initially classified it as a cave bear molar, not a human tooth, a bear tooth. That is how far outside the expected range of human dental anatomy these individuals fell. But what the Denisovans looked like is the least interesting part of their story. The genuinely astonishing part is where they went. And this is where the Denisovan puzzle becomes almost impossible to solve with the physical evidence alone. Denisovan genetic material shows up today in living human populations scattered across a vast geographic arc. If you are of Melanesian descent, Aboriginal Australian, Filipino Negrito, or from certain indigenous groups in Papua New Guinea, you carry between 4 and 6% Denisovan DNA in your genome. Some populations carry even more. A 2021 study, led by Maximilian Lorena at Uppsala University, published in Current Biology, analyzed the genomes of the Aita Magbukan people of Luzon, in the Philippines, and found Denisovan ancestry levels approaching 5%, among the highest proportions recorded anywhere on Earth. Read that map carefully. The one confirmed Denisovan cave is in Siberia. The strongest Denisovan genetic signal is in island populations of Southeast Asia and the Western Pacific. That is thousands of kilometers of ocean, jungle, and mountain range between the known fossil site and the populations carrying the most Denisovan DNA. Which means one of two things. Either modern humans interbred with Denisovans somewhere in mainland Asia and then carried that DNA southward as they migrated into the islands, or Denisovans themselves were already living in Southeast Asia and the Pacific region when modern humans arrived. Given how strong and how varied the Denisovan genetic signal is in those island populations, most researchers now lean toward the second option. The Denisovans were already there. They had spread across an enormous territory that included not just Siberian mountain caves but tropical forests, lowland coasts, and possibly islands that required crossing open water to reach. And that last detail, if confirmed, changes everything about what we assume the Denisovans were capable of. Crossing open water in the Pleistocene era means some form of boat or raft. It means planning, material construction, and navigation of ocean currents. It means a level of behavioral complexity that we have historically been reluctant to attribute to any human species other than our own. Those 4 to 6% numbers also tell a biological story that is easy to underestimate. That level of genetic contribution from a separate species requires sustained, repeated interbreeding over thousands of years across multiple geographic locations. This was not a single accidental encounter on the edge of a forest. This was generations of coexistence. Denisovans and Homo sapiens lived in proximity, formed bonds, had children, and raised those children together, for a very long time, across a very wide area. And some of that inherited DNA is not just a fossil relic of ancient contact. It is actively keeping people alive right now. In 2014, a research team led by geneticist Rasmus Nielsen at the University of California, Berkeley, published a study in Nature identifying a gene variant called EPAS1 in Tibetan populations. This variant regulates how the body produces hemoglobin at high altitude. In most humans, ascending above 4,000 meters triggers a dangerous chain reaction. The body produces too many red blood cells in an attempt to capture more oxygen from the thin air. The blood thickens. The risk of stroke, pulmonary edema, and organ failure spikes dramatically. Tibetans do not have this problem. Their version of EPAS-1 prevents the overproduction of hemoglobin, keeping their blood at the right viscosity even in desperately thin air. It is one of the clearest, best documented examples of adaptive gene flow ever identified. And the gene did not evolve in Tibetans through their own random mutations over time. It came from Denisovans. The Denisovans had already solved the problem of living at extreme altitude, probably across tens of thousands of years on the Tibetan Plateau and the surrounding mountain ranges. Their bodies had adapted to conditions that would incapacitate most modern humans within hours. When Homo sapiens moved into those same high-altitude zones and interbred with the Denisovan populations already adapted there, they inherited a ready-made biological answer to one of the harshest environments on the planet. No need to wait another 50,000 years for their own mutations to arrive. The solution was already written. They just married into it. Millions of people today can breathe and work and sleep comfortably at altitudes that would flatten most of us, because a now extinct species of human solved the problem first and passed the answer forward through their children. That gene did not come from our lineage, it came from theirs. If this is making you think, subscribe and turn on notifications so you never miss one. This channel only exists because of the people who share these videos, and I don't take that lightly. EPAS-1 is only one piece. Research published between 2019 and 2022 has identified Denisovan-derived gene variants in modern populations affecting immune system function, fat metabolism in cold environments, and aspects of craniofacial bone structure. Multiple studies have shown that some of these variants were positively selected over generations, meaning the humans who carried them survived and reproduced at higher rates than those who did not. The Denisovan contribution to modern human biology is not a footnote in our evolutionary history. It is built into the operating machinery of how billions of bodies function every day. There is also the water problem, and it is a big one. The populations with the strongest Denisovan genetic signal, Melanesians, Aboriginal Australians, indigenous Filipinos, all live on islands. Getting to those islands during the Pleistocene, even when sea levels were lower and land bridges connected some territories that are now separated by ocean, still required crossing stretches of open water. Crossing open water in the deep prehistoric past means some form of watercraft. It means planning, construction of a vessel from available materials, reading of tides and currents, and a willingness to leave sight of land. These are behaviors that, until very recently, most researchers were reluctant to attribute to any human species besides our own. If Denisovans were already living on islands in Southeast Asia and Oceania when modern humans arrived, and the genetic evidence increasingly suggests they were, then the Denisovans were seafarers. Not in the European age of sail sense, but in a practical, functional sense that implies cognitive and social complexity far beyond what a handful of teeth and one finger bone would ever suggest on their own. All of this, the genetic gifts, the geographic spread, the altitude adaptation, the possible ocean crossings, makes the absence of physical remains even more bewildering. We know from the DNA that the Denisovans were widespread, successful, and long-lived as a species. So where are their bones? One jawbone provides a partial answer to where they went, and it raises an even bigger question about what they were doing there. That jawbone was found in Baishia Karst Cave on the Tibetan Plateau, at an elevation of over 3,200 meters. A Buddhist monk discovered it in 1980 and gave it to Lanzhou University, where it sat in a collection drawer for nearly 4 decades. Nobody knew what it was. In 2019, a team led by Jean-Jacques Hublin at the Max Planck Institute confirmed through ancient protein analysis that it was Denisovan. The bone dated to approximately 160,000 years ago. DNA extraction was impossible because the specimen was far too degraded, but the protein signature was unmistakable. That result moved the entire conversation. It placed Denisovans on the roof of the world, at an altitude where the oxygen content is roughly 40% lower than at sea level. And they had been up there for a very long time, longer than our species has existed in most parts of the planet. The environmental answer for the missing fossils is straightforward. Most of the regions where Denisovans likely lived are catastrophic for bone preservation. Tropical forests, river valleys, and lowland coasts across Southeast Asia have the heat, the humidity, the acidic soils, and the microbial activity to reduce a skeleton to nothing within a few centuries. The cold, mineral-rich interior of Denisova cave is a geological accident, a natural deep freeze at exactly the right latitude with exactly the right chemistry to hold 75,000-year-old DNA intact. That kind of preservation is spectacularly rare. But there is a second possibility, and it is gaining traction. We may have already found Denisovan fossils sitting in museum drawers across China and been looking right past them. Across East and Southeast Asia, there are skulls and bone fragments that have confused paleoanthropologists for decades. Specimens too robust for Homo sapiens, too recent for Homo erectus, too strange for any established classification. They have been labeled archaic Homo and filed away, sometimes for decades, waiting for a framework that could explain them. The problem was never a shortage of bones in Asia. The problem was a shortage of categories that those bones fit into. And now, with the Denisovan genome in hand, the category exists. The question is whether the bones match. One of the most striking candidates is a massive cranium recovered from river sediment near the city of Harbin in northeast China. The skull was actually found by a laborer in 1933 during the construction of a bridge, hidden by its discoverer inside a well for safekeeping, and only donated to Hebei Geo University in 2018. It was formally described in 2021 and tentatively assigned to a proposed species called Homo longi. The skull has a brain case as large as our own, heavy brow ridges, and wide, square eye sockets. Several researchers have argued publicly that this skull and others like it scattered across Chinese institutions may be the Denisovan face we have been searching for. If that is true, the Denisovans were not a marginal, isolated population clinging to existence at the back of a Siberian cave. They were one of the most successful human species this planet has ever produced, occupying the largest continent on Earth for longer than we have had agriculture by a factor of roughly 50. And then there is Denny. In 2018, Pääbo's team at the Max Planck Institute announced a finding that rearranged the entire field. They had analyzed a bone fragment from Denisova cave, roughly 90,000 years old, and discovered that it belonged to a teenage girl whose mother was a Neanderthal and whose father was a Denisovan, a direct first-generation hybrid. Two separate species of humans had met in or near that Siberian cave, and they had a child together. That child's bone then survived 90,000 years in the sediment for a 21st century laboratory to find and decode. They called her Denny. She is the only confirmed first-generation hybrid between two archaic human species ever identified in the fossil or genetic record. Her mother's mitochondrial DNA traces back to a Western European Neanderthal population, hundreds of kilometers from that cave. Her father was Denisovan, but his genome also carried traces of much earlier Neanderthal admixture, meaning the two species had been meeting and mixing for a very long time before Denny was ever conceived. She was not the start of contact between Denisovans and Neanderthals. She was one visible moment in what appears to have been a sprawling, recurring pattern of interbreeding that persisted across tens of thousands of years and thousands of kilometers of Eurasian territory. And Denisova cave itself tells a story that the bones alone cannot. Sediment layers in that cave contain Neanderthal remains, Denisovan remains, and evidence of modern human presence, all in the same location at overlapping time periods. 3 different species of human used that cave, possibly at different times, possibly at the same time. The tools found in the cave include some surprisingly sophisticated artifacts, bone points and polished ornaments, in layers associated with Denisovan occupation. If those tools belong to the Denisovans and not to modern humans passing through later, then the Denisovans were making personal ornaments. They were decorating themselves. That implies symbolic thinking, aesthetic sense, and the kind of social complexity that we used to consider exclusively our own. For most of the 20th century, the story of human evolution was drawn as a tree with clean branches. One species replaced the next. Homo erectus gave way to later archaic forms, which gave way to Neanderthals in Europe and Homo sapiens in Africa. Our species left Africa, encountered Neanderthals, outcompeted them, and walked forward alone into the future. Clean, linear, easy to put on a poster in a biology classroom. That diagram is done. What the Denisovans, and Denny in particular, forced the field to accept is that the real shape of human evolution looks more like a braided river. Populations diverged, traveled separately for thousands of generations, met again, merged genetically, then separated once more. We did not replace everything we encountered. We absorbed parts of it, and it absorbed parts of us. The Denisovans disappeared as a distinct population sometime between 30,000 and 50,000 years ago. Nobody knows exactly why. Climate instability, competition with expanding Homo sapiens groups, gradual genetic absorption through continued interbreeding. Almost certainly all three, working together across millennia in ways no single fossil site can reconstruct. They left no cities, no cave paintings confidently attributed to them, no tool traditions that archaeologists can point to and say, with certainty, only Denisovans made these. But disappeared is doing a lot of work in that sentence, and it might be the wrong word. A species that vanishes by being absorbed into another species through interbreeding has not gone extinct in the way a species wiped out by a meteor has gone extinct. Its physical form is gone. Its distinct population is gone. But its genetic material is still out there, distributed, diluted, but operational. The Denisovans may not have died out so much as dissolved into us. Slowly, over thousands of years, one mixed child at a time, until the boundary between Denisovan and Homo sapiens became too blurred to draw. But they left something more durable than pigment on stone. They left their code inside living people. And that code is still running. Every Tibetan who walks easily at 5,000 meters is carrying Denisovan genetic information in their cells. Every Melanesian whose immune system mounts a specific defense against a specific class of pathogens is running on biological software that a Denisovan body refined across tens of thousands of years of survival and natural selection. The Denisovans are not gone. They are distributed, scattered across billions of genomes in fragments so small that no one noticed them until the sequencing technology existed to look at the right resolution. We spent a century assuming we knew the full cast of characters in the human story. Then a bone the size of a jelly bean from a Siberian cave added a character nobody had written into the script. There is a full chapter of our species' history that played out across the largest continent on Earth, persisted longer than recorded civilization by orders of magnitude, and left almost nothing behind you could see with your eyes. Almost nothing except the billions of people still carrying pieces of it in their cells right now, breathing air at whatever altitude their inheritance allows. And that is what makes the Denisovan story different from any other discovery in paleoanthropology. We did not find them in the ground. We found them in ourselves. The evidence was never in a museum. It was in a blood sample from a Tibetan farmer, in the immune response of a child in Papua New Guinea, in the genome of a woman in the Philippines whose body carries instructions written by a species that no living person has ever seen. The bones are almost gone. The signal is still transmitting. And the part that should sit with you long after this is over is how close we came to never detecting it at all. One discarded bone, one underfunded laboratory, one geneticist who refused to believe the past was unreadable. That is how thin the margin was between knowing the Denisovans existed and never learning their name. Thank you for watching until the end. If this changed how you see something, check out the videos on screen. I think they will hit the same way. And subscribe if you haven't. See you in the next one.